You're listening to Lecture 3. This is what you've been waiting for, FDA Drug Approval 101. I am actually doing this lecture because somebody wrote to me and said, when is Lecture 3 going up? And I said, you know what? That'll motivate me to get this done. So I'm Dr. Vinay Prasad. I'm an associate professor here at the University of California, San Francisco. I'm in epidemiology and biostatistics. I'm a practicing HemeOnc doctor. I work at the General, and I'm here to take you through FDA Drug Approval 101. This is part of my 10-part series. I've been doing this lecture for years. I used to give it to the Oregon Health and Science University fellows. Now I give it to you. I'm making it free based on advice of a colleague. So if you like what you hear here, I think you ought to read the book Malignant. It is a much better synopsis of my views. It is much crisper than whatever I say to you now, which is really extemporaneous speaking off the cuff. Every paper I describe on this episode, you will find at www.myname.com/papers. We got them all available. If you know what I mean, it's the place to look. If you know what I mean. We got a YouTube channel. We're trying to put out some oncology videos. Check out Plenary Session Podcast. I'm on Twitter for better or worse. Most days, that's for worse. That's for worse. That's for sure. Okay. And here are the papers. This is where you can find them. Everything we talk about, take a look. This is the series, Lay of the Land, Surrogate Endpoints, Control Arms, Crossover, Biomarkers, Drug Dose Reductions, Non-Inferiority, Genome-Driven Cancer Medicine, and Multiplicity, my favorite thing on earth. Where did we leave off? We've taken you through the lay of the land in episode 1. In episode 2, we're talking about surrogate endpoints. And here, we're going to give you the 101, what you need to know about the U.S. Food and Drug Administration. This is important stuff. This will tie everything together, I hope, by the end. So, terminology. There are a few vocabulary words you need to know if you want to be able to talk about this topic. 2, what about all those drugs approved on the basis of response rate? You'll remember in the last episode, we explained what was a response rate and the dinner party, as they say, the dinner party at Charles Mortel's house where we learned about response rate. Drugs approved on the basis of progression-free survival and drugs approved on the basis of overall survival or a patient-reported outcome or health-related quality of life, which I'm going to put in the clinical endpoints bucket. So let's go terms. NME, a new molecular entity. An NME is the first marketing authorization for a novel compound. So, the first time ibrutinib came to the market in CLL, that was an NME approval. A supplemental marketing authorization is the second time a drug is approved for some other purpose. If you take a drug like pembrolizumab, I think we're in the 30 club. There's 30 plus supplemental marketing authorizations. When we talk about a drug approval, what we really mean is a marketing authorization. It's the license to sell your product on the US market. That's what an FDA drug approval is. It's the ability to market your product. When we talk about an indication, so this is important, drugs are not approved or not approved. They're approved for indications. And an indication is this drug, given in a certain way, for this cancer, in this setting, by setting I mean the adjuvant or the metastatic setting, usually metastatic. Many, many more drugs are approved in the metastatic setting than the adjuvant. We'll mention that there. And what line of therapy is it approved for? And in combination with what other drugs? And only after you progress on what drugs? And only in what combinations? And all of that tied together, and any other sort of molecular features that go into it. That's all part of the indication. The drug, the setting, the line, and any extemporaneous sort of inclusion criteria for the approval. So we're talking about marketing authorization for indications. And an NME is the first one, and a supplemental new drug authorization is a subsequent one. So the other thing you need to know about vocabulary, you need to know accelerated approval. Accelerated approval means that the drug is provisionally made available to people. It's a conditional approval. The company is usually mandated to show some post-marketing signs of efficacy, to confirm efficacy post-market. That is the social contract of accelerated approval. We're going to let you come to market based on a surrogate endpoint, which I'll say in a second. And in return, we want you to prove your drug actually does something when it's on the US market. A regular drug approval, a traditional marketing authorization, this is the full approval. There can be some demands placed on the company, but those demands are not to fundamentally show the efficacy of the product. That's been established. So accelerated approval, a provisional approval where there will be a hearing someday, or the FDA will have to review your post-marketing commitment, and a regular traditional full approval where you get the marketing authorization and it won't be rescinded. Well, at least not for efficacy. Perhaps something crazy would happen and it would be rescinded, but it won't be rescinded for efficacy. Now, what about surrogate and clinical endpoints? A surrogate endpoint is, as my dear friend and mentor Adam Sifu likes to say, a surrogate endpoint is an endpoint the patient didn't know was important until the doctor told them it was important. So your hemoglobin A1C, you don't know it, you can't feel it, but when the doctor said you got to care about your A1C, suddenly it takes on a life of its own. LDL cholesterol. And in cancer medicine, surrogate endpoints are radiographic biomarker endpoints of progression such as M protein levels, scans, response rates, progression free survivals, disease free survivals, invasive disease free survival. These are all surrogate endpoints, major molecular response. The list goes on and on. And we always find a way to make new ones. We've got MRD coming. You're going to love MRD, all sorts of cancers. We're going to give you a new surrogate. We got PathCR. That's only been used once, but it is a new surrogate. What about clinical endpoints? Clinical endpoints are why we're in this business. Let's never forget. We want people to live longer or live better. And clinical endpoints are endpoints that show you that has happened. So those sorts of things include overall survival, the gold standard clinical endpoint. They also include things like health related quality of life, like patient reported outcomes. Now, patient reported outcomes, of course, it depends on what you ask the patient. And so you might focus on some outcomes and not other outcomes. And that might be a problem of some patient reported outcome scales. Health related quality of life, it depends on how well you do it. But overall survival is that one clinical endpoint that you really can't pull too many strings there. I mean, there's some things we're going to talk about in subsequent episodes, like post protocol care and misuse of crossover. That can distort an overall survival, but the endpoint itself is a real thing that we're measuring, and it can't be gamed by the type of questionnaire administered. So, I want to give you the lay of the land. You need to know a little bit about the lay of the land and how the FDA does approvals. And the best place to turn to is the paper by my dear friend Chul Kim, who's now an assistant professor at Georgetown University, a thoracic oncology specialist. But he was just a fellow when we did this back in 2016. This is the strength of validation of surrogate endpoints used in the US FDA's approval of cancer drugs. We looked at 5 years of drug approvals, and this is it. 5 years of drug approvals in one table. And I don't want to bore you with the names of the drugs, which you have likely already memorized. I want to talk to you about the buckets that these approvals fall in. So, one, there are some accelerated approvals and there are some regular approvals. And it's about a third and two thirds. One third of new drug approvals are accelerated approvals. Here we've combined NMEs and supplemental marketing authorizations. We're looking at all of the approvals in a row. Two thirds are regular approvals. Those are approvals that don't have post marketing commitments to show further efficacy. Accelerated approvals do, although they don't really like to enforce them anyway. That's something we've seen recently at the Oncology Drug Advisory Committee meeting. Surrogate and clinical. This is something people don't appreciate. Surrogate endpoints, they're not just for accelerated approvals. They can also be used for regular or traditional marketing authorizations. We'll go through what the statutes say. But two-thirds of drug approvals are on the basis of a surrogate. That means when they're approved, we don't know you live longer, and we certainly don't know you live better. Only one third is approved based on a clinical endpoint. So just to revisit this, accelerated and regular, two-thirds surrogate, one-third clinical endpoint, okay? You might think that they should have been two-thirds accelerated approval. And in fact, some people like myself would say that probably ought to be the case, but this is the way it is. We got two-thirds surrogate, one-third clinical, and we've got one-third accelerated and two-thirds regular. And now the final point, response rate and PFS. When you are an accelerated approval, you are almost always approved on the basis of response rate. And almost always that response rate was calculated in a single arm, uncontrolled study of people who are enrolling. That has some bias. There's a selection bias in the people going to that study. It's not directly comparable to anything because there is no control arm. Very seldomly, there's a response rate based in a randomized study that's used for response rate. I can think of one example off the top of my head. Very rarely do they give you an accelerated approval for a PFS benefit. We saw that with Avastin in breast cancer, for instance. That was an accelerated approval back there in, I think, 2008, 2009, based on a PFS benefit. Now, let's talk about regular approval. When regular approval is made on the basis of a surrogate, it's almost always on the basis of PFS. Although there are some instances where a response rate actually gets you the full regular approval, and there is no requirement for a post marketing efficacy study. So this is the lay of the land. There's some bleeding here at the edge of response rate and PFS. And then overall survival or patient reported outcomes is the final category. And it is mostly overall survival. Let's be honest, patient reported outcomes, quality of life, seldom seldom forms the basis of approval. This is the lay of the land. The FDA is steadily expanding categories where surrogates previously weren't accepted, and now they are accepted. They're expanding those categories as we speak. This is Emerson Chen, my dear friend, and now faculty member at Oregon Health and Science University, formerly a fellow there. And we did this paper, which we published in JAMA Internal Medicine in 2011 on the FDA's expansion of surrogate categories. And here's what this analysis shows. The blue dot is the first time a drug was approved for that indication based on that surrogate. So the first time, for instance, in relapse refractory mantle cell lymphoma, we used response rate as the basis of approval. That's the blue dot. And the green dot are subsequent approvals in that indication. Now, let's be careful what this isn't. This isn't an NME and supplemental marketing authorizations. Here, the categories are binned by indication. And so what you see is the first drug to receive approval in that indication based on that endpoint, and then subsequent drugs that receive approval in that indication based on that endpoint. Don't get yourself confused. Read the paper. Read the paper if you are. What you see here is we go all the way back to 1993, and we come all the way to the present. And I think what you see is what I see, which is the FDA is slowly and steadily expanding the tumor types for which we accept the surrogate. The more blue there is, the more different ways in which you can get an approval based on a surrogate. And in recent years, oh boy, they look eager to accept new surrogates, don't they? They look keen and eager to. And I think that is, in fact, representative of market forces. So when the FDA says we're using surrogates we have always used, that would be technically incorrect because every blue dot here is a new surrogate that is being christened for the very first time. So now let's talk. We also have in that paper a breakdown of accelerated and regular approval, but you can check that out. Let's talk about drugs approved on the basis of response rate, which, as we talked about in lecture 2, I believe, is tumor shrinkage beyond that 30% line in the sand. And why did they pick 30%? Well, that's what 16 men in 19 Diggity could tell apart with calipers through foam rubber. Obviously, that's why we would use the endpoint. It's the gold standard of what men in 19 Diggity could feel through foam rubber. That's obviously why we use it. And of course, why many people believe it's clinically meaningful because they don't know where it comes from. So when drugs are approved on the basis of response rate, there are 2 ways you can approve them. You can give it the accelerated approval. In this case, what you're saying is the response rate is, quote, reasonably likely to predict. The statutory language is clear. A surrogate used for accelerated approval means that surrogate is, quote, reasonably likely to predict clinical benefit, which means survival and quality of life, in my opinion. When a drug receives regular approval based on response rate tumor shrinkage, it means that surrogate is established. It's not just reasonably likely, like, we would reasonably think it would track with survival. It is established. Like, this is something you can hang your hat on. And we're going to talk about whether or not the FDA actually lives up to that statutory language. This is an analysis by Emerson Chen. Again, Emerson. I love Emerson because he was a fellow who worked under me, and I learned a lot from him. And maybe he learned a few things from me, and maybe some of them he can actually repeat. But a few he'll have to take to his grave. And we also work with Vikram, who's another fellow at OHSU. This is an overview of all the cancer drugs approved by the US FDA based on the surrogate endpoint of response rate. Now, I'm not talking PFS. I'm talking response rate, 30% or more tumor shrinkage. And we're looking at all those over a period of time. I believe 2006 to 2018, but don't hold me to it. I can't remember all these different papers. I remember the key things. I don't remember the dates. Here's a very nice figure. I like this figure. Emerson made it, of course. What he's showing you here is these are the response rates of the drugs that got approval based on response rate. This is what you knew about them when they were approved. And what you see here is that some drugs were great. Look at Imatinib, high response rate. Perhaps it was even higher. I'll have to go back and check that one. Tisagenlecleucel or Kymriah, 83% response rate. He might be plotting the CR rates or something more stringent here. No, he's plotting the regular response rate. 83% response rate for tisagenlecleucel. What's the problem here? Well, if you got your cells collected with a CAR T therapy and they didn't actually infuse it because something happened in the meantime, they couldn't make the product, or God forbid, the patient passed away, You should include that in a denominator. That's called an intention to treat denominator, but they excluded it for this one class of medications, which distorts, I think, the response rate upward. But yet that's what it is. And then you get all these different drugs. And here are the ones that are accelerated approvals that ended up being converted on the basis of survival. They proved a survival benefit. These are accelerated approvals that became converted to full approvals to get the regular approval because they later showed they improved progression-free survival. These are accelerated approvals on the basis of response rate that were converted to full marketing authorization on the basis of response rate. Now, this is a paradox that a number of commenters have pointed out, which is you're saying response rate was good enough for an accelerated approval. And then to prove that you had clinical benefit, you accepted more response rate, but response rate was already deemed insufficient for regular approval at the outset. So how did it become so good after all? And the answer is this is a deep paradox and it's problematic. And I would even say this yellow is problematic too, because I don't want drugs that improve response rate to prove they improve PFS. That's just a different surrogate. I want them to prove that people live longer. The blue, or sorry, not the blue, the green. The blue were drugs that got regular approval where there is no post-marketing commitment. There, of course, I also want them to convert to green, and they don't always do so either. And then the purple are drugs that got accelerated approval because they had a response rate, but they have never yet proven any other endpoint, have not been converted to full approval. And the dotted line shows the median response rate. And what do I want to say? I think drugs that get approval in uncontrolled studies are probably the most active drugs. That's why they're getting the approval in uncontrolled studies. They have a 40% response rate, which I think is actually pretty decent as an oncologist. However, they don't have a 99% response rate. They don't even have an 80% response rate. We can talk all you want about the ALK inhibitors, but they don't represent the typical approval based on response rate. And some of them are rather, I think, quite lackluster. I mean, look at over here, these sort of tails. This is not good. 12%, 13%, 14% response rate. No, I don't like that. That's not good enough. I need show me some randomized data that you're better than comparators. Higher response rates, maybe we would tolerate some of those approvals, but you still got to do some post-marketing studies to show you're better than alternatives, I would imagine. Here's the CR rate graph. And I've plotted the phase 1 study of Imatinib just to put it all into context. Oh, I know why the other figure. It wasn't Imatinib in CML. It was Imatinib in a different malignancy, maybe GI stromal tumor, maybe something else, maybe something else, but it was still pretty high. This is Imatinib in CML. And these are the complete response rates in all the other tumor types that got approval based on response rate. What do I see here? I mean, we can talk about Imatinib and people can say, like, look, when you got a drug as good as Imatinib, you can't randomize, it's difficult, equipoise, parachute. They can say all that usual stuff people like to say. But what I would say to you is your other drugs ain't no Imatinib. The median CR rate for the other drugs is 6%. You mean to tell me 6% is like 98%? Get out of here. Not even close. And so, the regular approval drugs had higher CR rate. These had this CR rate. These had this CR rate. But there are a lot of low CR rates. I mean, these are not drugs that are so potent in generating CRs that one can rest assured that, survival is guaranteed, because you really need a durable CR that lasts for a heck of a long time like Imatinib does. So in other words, drugs approved based on response rate, they ain't no Imatinib. They ain't no Imatinib. That's what I have to say. This is a great paper by Alison Haslam, who works with me. I give her a lot of credit. She did a really nice paper here. It's called The Response Rate of Alternative Treatments for Drugs Approved on the Basis of Response Rate. Whoa, what does that mean? Alison does something quite simple. On one axis, the x-axis, Alison is saying, what's the response rate of a drug that was approved on the basis of response rate? She's basically plotting Emerson's figure right here on the x. On the y-axis, Alison does what many people don't do. She hits the books. She reads about these indications. And she says in each of these indications, before this drug was approved, before this new mantle cell drug in the relapse refractory setting with a response rate of X, what was the response rate of a drug we used to use in that setting? Some old-fashioned chemotherapy drug. It's not on patent. No one's excited about it. What was the response rate of those drugs? And she plots that on the Y. And the red line is a 1-to-1 line. It's a 1-to-1 line. So what do I want to say here? If people wish to claim, And they do. Trust me, they do. If people wish to claim that the new drugs we're approving have response rates so much better than the old drugs that it would be wrong to test them in randomized controlled trials, we don't have equipoise. They're so much better. We know they're better because their activity is higher. That's what response rate is, a surrogate endpoint, a measure of activity, not of clinical benefit, of course. But were they to say this, all of the points would be well below the red line in the lower right-hand corner of the figure. What Allison shows you is that many points are scattered around the line, suggesting that there is a lot of equipoise with pre-existing treatments. And what Allison shows you is that if you're not on the line, you're at least close to the line. And equipoise doesn't just exist for being on the line or above the line. It's anything close to the line. Because you know what? Medicine can be surprising. A medicine doesn't always go the way you think, especially in randomized controlled trials. So I think this figure actually debunks the idea that we have to give accelerated approval based on response rate because we lack equipoise, or we have to give regular approval based on response rate because we lack equipoise, because in many cases, we have older options that are just as active or potent or generate as high response rates. And we don't know which is better in terms of maximizing survival or quality of life, which, let us not forget, is actually why we're in this business. Let's not forget that. So those are the drugs approved based on response rate. Now, let's take a closer look at the drugs approved based on PFS. Now, remember, these are generally, quote, established surrogates. They're established. They're established. How much more do you need? Established. We'll talk about that. So the slides in gray mean that I think I've previously shown them to you in a prior episode. So go back, double check. This is, of course, a figure by a paper by Joey Del Pagio, led by Chris Booth, that I'm lucky to be a part of in HemeOncology. Shows you that overall survival used to be the primary endpoint of randomized controlled trials in this business de jour in the heyday of the 85 to 94, back when oncologists were giants. But of course, it has diminished in popularity and been replaced by progression-free survival. Strangely, that went along hand in hand with funding from the biopharmaceutical industry. It's almost as if the industry realized that OS was so unattainable that PFS would maximize their market share that they pushed us towards PFS as the primary endpoint. But that couldn't possibly be the case. Also from lecture 2, how do you validate an endpoint? Now, remember, I talked to you about trial level validation. And so we imagined all of the randomized controlled trials in a space. We plotted them on an axis. There was a delta PFS, a delta OS. You could have replaced that with hazard ratio. You perform linear regression. The way in which the points correlate, cluster on the line, tells you something. The R or R squared tells you about how strong or valid that surrogate validation is. This is literally asking the question, what percent of the variability in the endpoint you care about, OS, is captured by variability in the surrogate, PFS, in randomized prospective controlled trials of novel anti-cancer drugs? That's literally what it's asking, literally what it's telling you, and it is the most important study, trial level validation. You can check out our paper in 2015 in JAMA Internal Medicine. We talked about IDFS or DFS and adjuvant breast cancer. I showed you an example of the figure. You saw 2-year DFS on the X and 5-year OS on the Y, one metric of the relative difference. Each dot is a randomized controlled study. The size of the dot is proportionate to the sample size of the study. I showed you the R was 0.62, which is weak. IQWiG from Germany would say unsuitable for regulatory decision making. And I showed you that most of the surrogates we're using are quite weak. The blue is weak in the metastatic setting, weak, or medium, which is also considered unsuitable for regulatory decision making. And just a few were good correlations. Finally, I showed you some work by Chul Kim and I that appeared in JAMA Internal Medicine many years ago, where we followed these approvals based on a surrogate into the future, be it regular or accelerated, and we followed it out into the future, and we asked what percent had studies that confirmed a survival benefit, and it was the paucity, a tiny fraction, and most did not. Most did not have those studies and a 4.5 years follow-up. And I said, and I looked into the camera and I said this, I said, I'm willing to accept drugs that come on the market. And I don't know if people live longer or live better as a result of having those drugs, but I will be damned if I'll accept the fact they can go their entire life cycle and make tens of billions of dollars, and we still never know. I think we have to say, at some point in the life cycle of the product, we have to know. Maybe you can drop $1 billion on it before you need to know, but when you start to get to double-digit billions, I'm going to say, hey, does this actually work? And don't call me crazy for that. I think that's quite reasonable. We talked about quality of life. Drugs approved on the basis of the surrogate in the paper by Tracy Rupp, they don't improve quality of life necessarily in the future. So back to that paper by Chul Kim, what is this reasonably likely to predict? What is this established? What does this mean? And that's what we took a look at in this paper. This is contraband. This is forbidden stuff. This is a slide that slipped out from the FDA. And this is from the FDA. They presented at some workshop. And here's what they say. For accelerated approval, because the regulatory standard is, quote, reasonably likely to predict, surrogate validation is not strictly required. You just have to believe in your heart that that surrogate is a reasonable surrogate. I think that's quite weak, but I'm willing to take it. For regular approval, to replace a surrogate or intermediate endpoint for clinical benefit endpoint, validation is necessary. Okay, I like that. Trial level surrogacy, for example, an R squared of 0.8 between the treatment effect based on surrogate endpoint and treatment effect based on clinical benefit is needed. So they're saying R squared 0.8, that trial level validation, exactly what I showed you. I like it. I think that's a fair threshold. I think rather consistent with German IQWiG. Back to our figure. You remember, we looked at all the drugs. So let's look at the strength of the surrogates. We've got the drugs in the leftmost column. These are mostly response rates, 1 PFS, used for accelerated approval. We have the drugs in the middle column. This is mostly PFS, but some response rate, used for regular approval. They must be established. And we have the drugs that have already proven OS or quality of life benefit, albeit in studies perhaps with limitations. Future lectures, my friends, future lectures will come to column 3. Let's look at the accelerated approvals. Right here should have been a bar, but it's missing. And that bar that's missing is the percent of drug approvals, FDA accelerated approvals that use a surrogate with a proven established high-level correlation, that R squared of 0.8. It doesn't exist. How many use a medium strength correlation? And it don't exist. How many use a low correlation in a trial level validation study? And it don't exist. And how many used level 1 with low correlation is the brown. There's a few that have a low correlation. I see 2 bars are missing, not 3. How many had a level 2 study? This is a study that doesn't show that it's a surrogate. Rather, it shows that it is a prognostic endpoint. And the answer was some of them, but most of them had no study. You could look in the literature all you want, but you would never find anyone who's ever asked if that surrogate response rate typically had anything to do with anything in that tumor type ever. So we just don't know. It's really a gut feeling. This means most accelerated approvals are based on a surrogate that somebody said, I believe in it. And that's as sciencey as it got. Scienceism. Now, here is, and I believe my face is mucking this up, but here is the regular approval. We do have a few, 3, that actually meet the statutory requirement of an tight R squared in a trial level validation. We've got a bunch that fall below that. They've got a level 1 study. They have a trial level study, but it had a medium correlation. Here at level 1 and level 2 means trial level or something else, which I won't bore you with to make this lecture a little bit easier. The brown bar are trial level validation studies that have a low correlation or poor correlation. The yellow bar is a prognostic study, and the red bar is they don't have any study at all. And what you see here is rather clear. It's rather sobering that although that FDA slide said they wanted only orange bar, what you find is very little orange bar. So I believe that the FDA is in fact not living up to its stated regulatory standard. It is approving drugs with regular approval that have evidence beneath their stated standard, and that is a deeply problematic problem in the field. And it's not enough. It's not enough for them to just approve drugs all the time based on surrogates. We have no idea if it correlates with living longer, living better. They go one more, and they find new surrogates all the time. So this is the story of pathologic complete response. If you want to know the full story, I'll direct you to the Journal of Cancer Policy, 2018, a few years old. Jia Lu, who's now at Memorial Sloan Kettering, and myself, we asked the US FDA's use of PathCR as a regulatory endpoint. Did it pay off? Did it pay off? That was the question. I've used these words a little bit. Let me be more precise. You need to know the difference between prognostic, predictive, and surrogate. And they're a big difference. So prognostic endpoints tell you how a patient's going to do in the future. 17p deletion, they're not going to do as well as if they had p53 working well. It tells you how they're going to do irrespective of treatment. A predictive endpoint, HER2 in breast cancer, that tells you which patients benefit from some therapy versus which do not. Predictive endpoints can also be prognostic. Of course, we know HER2 is an adverse prognostic feature, but predictive endpoints more to point. They tell you that if you're HER2 positive, you benefit from certain classes of drugs that simply don't work if you're not HER2 positive. It is a predictive endpoint, predictive biomarker. A surrogate endpoint or biomarker tells you that drugs that impact this biomarker are also likely to impact things you care about. It really is the regulatory hurdle and question, and it really does require that special type of validation that I've described to you now twice with that scatter plot. This is neoadjuvant breast cancer. This was Lapatinib, trastuzumab, and the combination of HER2-positive breast cancer in the neoadjuvant setting. What does that mean? They give you the drug, they do the surgery, they look under the microscope. If they can't find tumor, you've got a path CR. If they can, you haven't. And what they show you here is rather crystal clear that you take one drug, you have some path CR rate. You take the other drug, you've got some path CR rates, but you take both drugs and you got a good path CR rate. You're doing really good. Path CR, of course, got to be good, right? You cut out the cancer and there was no cancer left because the drug works so well. Got to be good, right? Got to be good. And in fact, in the FDA's own analysis by Pat Cortazar and colleagues that appeared in the Lancet Oncology, which was the basis, in fact, it helped pave the way for pertuzumab to gain approval in the neoadjuvant setting. In the Cortazar analysis, Pat Cortazar found that women who achieved PAT-CR, the red line, they had better OS than women who didn't achieve PAT-CR. So one, I showed you the combo increased PAT-CR rate. Two, I'm showing you data. The better your PAT-CR, the better the survival. Hey, whoa, this is good. This is a surrogate endpoint, is it not? Let's be careful. It's actually proven to be prognostic. Pat Cortazar has proven it's a prognostic endpoint. Has she proven it's a surrogate? And later in her same paper, figure 6, she actually did the right analysis. These are the odds ratios of PAT-CR, one arm to the other in randomized controlled trials. This is the hazard ratio for EFS or OS. We really care more about the figure on the right, the OS, not the EFS. But either way you slice it, the line is flat. I don't know if you know this, but if you do a linear regression and the line is flat, the R is 0. There's no relationship between the variables. It's all over the place. There is no relationship between these variables. It has nothing to do with it. PAT-CR did not predict an improved hazard ratio of OS. It's not a surrogate at all. It's a prognostic marker, but it is a failed surrogate by their own analysis. But that didn't stop them. Don't let that stop you from approving pertuzumab. And in fact, they did. Well, they also said it had a big benefit in the metastatic setting, but that's also neither here nor there because drugs that have worked in the metastatic setting haven't always worked in the adjuvant setting. We'll talk about that in a future lecture with a paper by Eddie Maldonado that we did. Why does this happen? How is this possible that PAT-CR is good, not having PAT-CR is bad, but a drug that had a higher PAT-CR rate doesn't necessarily mean you live longer? And I think you have to think about individual patients. So on the left, this is the world where you didn't get the drug, or sorry, you got one drug. There's some people who didn't achieve PAT-CR and some people who did. Let's say the red patients didn't achieve PAT-CR, the blue did. Or to make it match the other figure, let's flip it around. The red did achieve PAT-CR. They're doing better than the blue. Let's go back. Yes, the blue are the ones not doing as well. The blue didn't achieve PAT-CR. The red did. And in the other one, you got more people with blue, more people achieving PAT-CR. Oh my God, these colors are backwards. Okay, last attempt. The blue squares mean that you have achieved PAT-CR. The red mean you haven't. Red is bad. That's how I intuited that. So I'm going to ignore Pat Cortazar's colors. Here, you've got a higher fraction of people with PAT-CR. So you would predict the group on the right's going to do better. But the real question is this. These people who are blue, the whole difference in the outcome is going to be driven by these 3 people. Are these 3 people, I've shaded them in purple, are they going to behave as favorably as the people who achieve PAT-CR with less drug? Or are they achieving PAT-CR because you're pushing drug on them? But that PAT-CR is not a robust PAT-CR. It's something flimsy. It's something ready and ripe to recur. Are they really going to behave more like the red patients? The real question of the surrogate is the people who flip, who would have had one outcome and now have a different outcome on the surrogate. Is their outcome so much better that the entire curve does better? That's the question. And in fact, we've seen the answer in this business. This is ALTO. This is the adjuvant study of Lapatinib plus trastuzumab versus one or the other. And what they find and show rather clearly is there is absolutely not a lick of benefit in DFS, even though the combo does generate a higher PAT-CR rate. So this should have given people a dark feeling in the pit of the stomach. We had approved pertuzumab. We didn't find a DFS benefit for another combination that improved PAT-CR. Pertuzumab plus trastuzumab improved PAT-CR in the neoadjuvant setting, but would pertuzumab succeed in the adjuvant setting? And we did get the result. And this was different because this is a P of 0.045, and that's a win. That's a win. But look at the curves. Look at the curves. Boy, you can say a lot of things, but I think one thing you can't say, you can say that pertuzumab has a role in adjuvant breast cancer. You can say for somebody with maybe 3 nodes plus, for somebody with a big tumor, with HER2 positive tumor, it still has a role, maybe in that cohort. You might even want to use it more widely. But don't look at this data and tell me PathCR is a good surrogate, because it ain't a good surrogate. Because pertuzumab plus trastuzumab cranked up that PathCR. And pertuzumab, when given in combination with trastuzumab, barely did anything to DFS. And we don't know anything about OS, but I'm not holding my breath. So it is not a good surrogate. It is a failed surrogate, but it shows the FDA's appetite for risk and developing new drugs and benefiting their prime constituency, the American people. No, I mean the companies, of course. It's the companies, the prime constituency. So, last example of this talk. I've gone too long. Neratinib. Neratinib is a small molecular HER2 inhibitor, of course. We all know that. To my knowledge, that's what TMK means. The only FDA approved therapy approved in the adjuvant space before the metastatic space. We're going to come back to that in a future episode of this series. But it really did. It triumphed in the adjuvant space before the metastatic setting. That is unusual. That's not the path drugs usually take. When you give 1 year of adjuvant neratinib after 1 to 2 years of trastuzumab adjuvant therapy, this is the IDFS benefit. And as they say in oncology, if you can fit a laser pointer between the curves, you can give the plenary session at the national meeting. And in fact, you can with a steady hand, the steadiest hand on earth, you can fit that laser pointer right between those curves. So it got regulatory approval, of course. Well, that's a spoiler, but we'll come to that. It's a toxic drug, of course. It had grade 3 toxicities, 41%, grade 4 toxicities over placebo. I'll show you the figures here. I think we forget that grade 3 toxicity for a TKI taken daily is not palatable. Here's what it means. It means an increase in 7 stools per day over baseline. You're going to the bathroom once or twice a day. Now you're going 9 times a day. That's a life-altering event. Your whole life is revolving around thinking about where the next restroom is. It's really a catastrophic side effect. Diarrhea requiring IV fluids hospitalization, interferes with ADLs. This is not good. And grade 4 is life-threatening complications. So this is the toxic drug. No ifs, ands, or buts about that. I always do this in lectures, and someday I think I'm going to scoop myself because I'm showing you in this lecture, somebody's going to know. I always show these 3 Kaplan-Meier curves, and I say, these are 3 trials in breast oncology. One is a drug, one is neratinib, one is pertuzumab, and one is, of course, a surgery, axillary nodal dissection that we no longer do for people with ER, PR positive tumors that have a sentinel lymph node positive, because we have done this randomized trial called Z11. And what you see here is one is a negative study. There's no difference between those curves. 2 are positive studies. And I ask people from the audience, is A, B, or C the negative study? Which is the negative study? And most of the time they say B is the negative study. But of course, B is, you've seen it already, it's pertuzumab, the adjuvant study. A is, of course, neratinib. C is the negative study. C is the study that the difference between the arms is not statistically significant. And C is the study why we don't do axillary nodal dissection. It's Z11. So I ask people, why is C a negative study, but A and B are positive? What's the reason? And occasionally somebody in the audience gets the answer, which is sample size. The sample size of A and B is in the thousands of people, and the sample size of C is in the hundreds of people. And what I want to point out is the deep problem in breast adjuvant setting, which is if you are running a clinical trials agenda where your positive trials and your negative trials are not separated by the magnitude of the delta, but if they're only separated by the sample size, you are running a perverse agenda. You're basically saying that any for-profit company that's willing to shell out big bucks and put a lot of people on your study can eke out whatever statistically significant difference of whatever not clinical meaningful difference that you wish, but that cooperative groups that seek to do other studies that don't have that kind of budget, they will have null findings with the same deltas. And that to me is deeply problematic. And I think the field of breast oncology should do some soul searching to ask if their trials are in fact overpowered, which I believe they are. So Neratinib, IDFS. Of course, it was approved based on that sliver of IDFS. I told you I could fit the laser pointer between those curves. And what would that IDFS translate into? Of course, we don't have an IDFS plot. This is a DFS plot, but it's the same surrogate validation study. I show you where Neratinib falls. It falls right there, 2.3% increase in IDFS at 2 years. That translates into an OS benefit of anything from -5% to +7%. So anyone who wishes to tell me that Neratinib is a guaranteed improving someone's survival, I would argue with you that there is no credible evidence that that DFS benefit will translate to an OS benefit. So what do I know about Neratinib? I know this. It's a toxic drug. Women are going to the bathroom 8 times a day. It's a life-altering event. It's a deep toxicity. Now, the company will say that it can get better with some supportive care, et cetera, et cetera. You can say whatever you wish. This is the result of the clinical study. There's a marginal improvement in IDFS. It's a surrogate that it has a poor correlation with OS. The magnitude of your IDFS benefit is so marginal that it's unlikely I have any confidence that you will translate to an OS gain. So there's no proof that surrogate will give me a greater survival. So what does the FDA do? What did the company do? They do this. Puma is expected to price the new drug, Neratinib, at a higher than expected 10K per month. This was back a few years ago. So disappointing. I think this is why our field is really suffering. It's a field that increasingly has substituted good clinical care for the development of largely financial products that purport to offer better health. The last thing I'd say to you is surrogates and speed. You know, people always say this to me. Well, the reason we're so in love with and addicted to surrogates is that they speed the time of results, and every day matters if you're somebody with cancer. And I don't deny that. In fact, I know that to be true. I take care of cancer patients. Every day does matter, and I'm always waiting for new drugs. But I don't want just more options. I want more good options, drugs that make my patients live longer and live better. And my patients often want more information. But with Emerson Chen, again, we sought to quantify the speed that you save when you use these surrogates. Surely it is massive, surely, because that's why we're using the surrogates all the time in 2/3 of drug approvals, as I told you. And here's what Emerson finds, quote, using surrogate endpoints in trials to get cancer drugs to market shaves 11 months off the average 7.3-year development time. We performed a meta-regression study, and we find that drugs take about 7 to 8 years to come to market, and you shave about 11 months off of it. You're not shaving much more than that. Now, why is it the case? Well, let me show you why. If you do a randomized control trial, you're measuring the primary endpoint from the moment people enroll in your study. You're measuring survival. If they die at any time, their scoring is an event. If you measure response rate, you've seen these kind of spider plots, plots that show you the depth of tumor response over time. And it takes a while before somebody might cross the -30. It might take weeks. You've seen it. You'll agree. You've seen these kinds of plots. What these plots don't show you is that the patients who respond and those who don't respond, they didn't all come to your clinic on day 1. That's an artifact of the plot. Some of the people who didn't respond came on day 17, and some people who did respond came on day 1, or the other way around. The first few people didn't respond. They came early, then people responded. So now the time until somebody responded is actually quite long. It's the time until they responded plus the time they enrolled. And if you start to think about it, the time it takes to yield a study result, it's not the time to see a response in an individual patient. In an individual, response comes first, progression comes second, death comes third. Typically, that's how we think about it. But this is a trial, it's an aggregate of people. And in an aggregate of people, you enroll people in a period of time, the enrollment duration, and there's a certain time until you have enough events that you can score your primary endpoint. And that cumulative time from the date of the last enrolled patient to the primary endpoint analysis, that's the time that the endpoint is taking, right? So we have looked at this, and it turns out that when you enroll people, there's some time to observe the median response rate, and then you have to observe something else. Because the FDA doesn't just approve drugs that generate response, they want to see those responses don't vanish immediately. They often want to see the median duration of a response. They want to know that the drug generated response in a fraction of people. Who knows where that occurred? If the drug generated response in 1 in 10 people, that might have been a lot of people at the tail end of your enrollment. You have to wait until they responded and then wait to see when half of them stop responding. That's how you find the median DOR. Or at least wait to see that the response is long lasting, 20, 30 months, etc. And what that means is it's all adding time. There's the enrollment, the time to observe the median response rate, and the time to observe the median DOR. Meanwhile, if you had done the enrollment, done a randomized study, you might have already been finding the endpoint as people passed away. You don't have to wait for responders to respond and then to progress, which is very long, actually, it turns out. And so what we find is when you do a meta regression and you factor in these things, that time savings actually is diminished quite a bit and it comes to 11 months. The last thing I'll say, this is a very provocative idea. I wrote this with Robert Kemp from Oxford Medical School, and we wrote this in a paper on surrogate endpoints. And it's also, I think, featured quite nicely in the book Malignant. But imagine this scenario that surrogates do more than change an individual trial. They change the trials that are even run. So imagine a drug company in a world without surrogates. In a world without surrogates, a drug company's got a new drug in breast cancer. They want to go in the relapse breast cancer setting because the drug company, because the FDA ain't accepting surrogates, and they want to show you they have an OS benefit in 12 months. This is in the world where we don't accept surrogate endpoints. People think people think and what people do is different. Okay. So people think if you allow surrogate endpoints, the company was going to run the frontline breast cancer study. Instead of waiting 30 months, you wait 15 months, the red arrow. People act like taking surrogates just does this, speeds the time up. But what surrogates do, it also changes the trials the company runs. Instead of running this trial in the last line setting, the company chooses to run this trial in the frontline setting. They barter the speed of the surrogate for market share. They barter this because you have a much bigger market share. So I think having an FDA that accepts so many surrogates can be deeply counterintuitive. One of the things that can lead is the paradoxical market incentive that companies now have a path to market and a broader market share. So they may actually seek to do different trials, trials that try to bite off more market share that roughly conclude in the same period of time using the surrogate endpoint. So surrogates may not even speed time, they may even elongate time. The speed may be bartered for market share. All right, the last part, drugs approved based on OS. We're going to hear more about this in future studies, but of course, you remember these drugs, 2.1 month average benefit, they are marginal. And there are a bunch of reasons why those marginal benefits are, I think, distorted. They're upwardly distorted because of the choice of control arms, because of the use and misuse of crossover, because of the lack of appropriate per protocol care, and because of drug dose reductions. And also, a few other things, which we're going to come to. We're going to come to. We're going to come to in the future talk. So, on that positive note, if you enjoyed this lecture, based on listener request, check out the book Malignant. You can listen to the YouTube channel. You can check out Plenary Session. Go to Twitter at your own peril, and get all the papers right here. So, yep, I think that's a wrap. In future episodes, we're gonna do all those good things. So many good things are coming. Multiplicity, my absolute favorite. You won't want to miss this. You will not want to miss this. So that's all I got for you. Thanks for sticking to this talk.